Hi, welcome back to our part 2 of chapter 24, your digestive system. So let's continue with the regulation of the digestive functions by 3 different types of control. Again, there's always 3 ways to control and regulate your digestive functions: local factors, neural control, and hormonal control. Local factors such as pH, volume, chemical composition of intestinal contents can affect the digestive system. So, for example, if chyme has moved into the intestine and caused your intestinal wall to stretch, this will stimulate the contraction of its smooth muscle to move things along and progress along your digestive tract. Or if histamine was released by your stomach cells, this will lead to secretion of stomach acid by those parietal cells we talked about. So for neural control, we have the mesenteric plexus. Remember, plexus refers to a network of neurons. For short reflexes, remember, short reflexes means it does not require the central nervous system in this type of reflex. It is just happening locally, controlled by the cells. So this is what activate peristalsis and segmentation within just that area of the small intestine. So for example, if you have your small intestine right here, there's your little bit of chyme, and around this area is where you will have short reflexes that tells it to move things along. Now, long reflexes are those that does require your central nervous system, and you have sensory information going toward your central nervous system. It will trigger that something that involves a bit more like a large scale movement and peristalsis of materials from one region of the digestive tract to another. Okay, so it's for large and farther distance of movement. And lastly, you have your hormonal control, which are achieved by enteroendocrine cells. Enteroendocrine cells. These are those that are located in the epithelium of the digestive tract. These cells will release a variety of hormones that enhances or diminish sensitivity of the smooth muscles to neural commands. We will talk about these 3 types of responses in application to our gastric activity next. So there are 3 phases in regulation of gastric activity. First phase is called the cephalic phase. This is when we are preparing for food's arrival. For example, we're thinking about food, smelling food, reading the menu at the restaurant, or seeing food pictures. Those are what's going to trigger the cephalic phase of our gastric system. So the parasympathetic division will be activated by the vagus nerve. Remember, this was a cranial nerve from AMP1, where your mucus cells, chief cells, parietal cells, and G cells, all those cells we mentioned in the stomach, will be activated to produce their specific gastric juices. This effect can be exaggerated or even inhibited based on a person's emotional state. For example, when someone is angry, stressed, or anxious, depending on the individual, someone may feel extra hungry in those cases, or others may feel the other way around, where they completely lose their appetite. It can go either way. The second phase is called the gastric phase. This is when food actually enters into the stomach. So your stomach is expanding, so that, sort of triggers the receptors for that. An increase in pH will be sensed as well due to arrival of food. The local response in this phase is where the expansion of the stomach will cause histamine to be released. Okay, so This histamine will then bind to parietal cells and signal them for release of stomach acid. Okay, so this is the parietal cell, which will then release hydrochloric acid under gastric phase. The neural response is where stimulated stretch receptors, okay, those stretch receptors right here, will sense the expansion of the stomach. Chemoreceptors will then activate secretory cells and mixing waves in the stomach to start mixing and mashing those digestive juices. The hormonal response is where the presence of protein in the chyme will stimulate secretion of gastrin right here. So remember, gastrin is secreted by G cells, which will then further activate your parietal cells and chief cells and your mucus cells. Those are all important for release of gastric juices that will be critical for digestion purposes. So all of these will combine, your parietal cells especially will release acid to reduce the pH level, and your chief cells will release proteolytic enzymes that will start digesting the proteins. The third phase of this gastric activity regulation is called the intestinal phase, and as the name suggests, this is when chyme enters the small intestines at a well-controlled rate so that we can ensure all your secretory, digestive, and absorptive functions will proceed efficiently in the intestine. So the neural response is where now you have less stimulation of stretch receptors in the stomach because it's being emptied, but there's an increased stimulation of stretch receptors in the small intestine. So here's your stomach decreasing in size due to decreasing levels of chyme within the stomach. But those in the small intestine are being expanded because now chyme is entering into the small intestine. So both of these actions will result in what's called the enterogastric reflex. Make sure you remember what this enterogastric reflex induces. So this is when gastrin secretion is inhibited. So when gastrin is inhibited, it inhibits both your chief cells and parietal cells from further secretions of acid and their enzyme. And then you have your pyloric sphincter here, okay, right here, pyloric sphincter will be stimulated for contraction to ensure that whatever entered into the small intestine stays in the small intestine and doesn't try to backtrack into the stomach. The other thing, when gastrin secretion is inhibited, it will also cause peristalsis to be inhibited as well. Okay? All right. So as the lipids and carbohydrates arrive in the small intestine, this will trigger the release of two important hormones, cholecystokinin and gastric inhibitory peptide. So these are called CCK or GIP. Both of these hormones will be important for their particular functions that I will talk about in a bit. Now, because chyme was mixed with the stomach acid, its low pH stimulates release of another hormone called secretin. And then again, like I said, we will talk about the functions of these hormones and what organs release them in a bit. Now, there are two central reflexes that are also triggered by a full stomach. You have your gastroenteric reflex, which stimulate motility and secretion along the entire small intestine. Make sure you don't confuse this with enterogastric reflex. They sound similar, but they are not the same thing. second reflex is called the gastroileal reflex, which allows movement of materials from the small intestine into the large intestine by opening what's called the ileocecal valve. So the ileocecal valve is easy way to remember by its name for the location of this. It is between the ileum, which is the last section of your small intestine, and the first region of your large intestine, and hence the valve is somewhere between the ileum and the cecum, and hence its name, ileocecal valve. Okay? Now, let's talk about some of the accessory organs of the digestive system before finishing off the last two parts of the alimentary canal. The pancreas, remember, we talked about its endocrine function in the endocrine system chapter, where cells within the pancreatic islets were producing insulin and glucagon. We mentioned before that the cells around those pancreatic islets are then called the acinar cells. These cells are responsible for producing pancreatic juices that will be released into the small intestine through the pancreatic duct. So here's the pancreas, and in the middle of it is what's called the pancreatic duct. And the pancreatic duct will then drain into the first section of the small intestine called the duodenum. Okay, duodenum. That's the first section of the small intestine. And it does so by, here's that pancreatic duct. Okay, and then it will drain through what's called the duodenal ampulla, sort of an expanded region right there, and then it will be released into the duodenum through a small opening called the duodenal papilla. So from the pancreatic duct into the duodenal ampulla, and then out of the duodenal papilla, which is this dot right here, and release the secretion into the small intestine, duodenum, the first section of the small intestine. The pancreatic juice contains this alkaline mixture of digestive enzymes, water, and ions. Now, earlier we mentioned that in the intestinal phase, secretin is the hormone that is released by cells lining your small intestine, and that it stimulates the pancreas to release a buffer containing bicarbonate ions and phosphate. Those ions will then neutralize the stomach acid in the chyme. Remember, the inner lining of the stomach has protective measures against its own acid, and hence that's why we were saying their mucus cells has to be replenished, and it releases a lot of mucus to prevent the stomach acid from ruining or damaging the walls. But the small intestine, however, doesn't have that protective measure. And hence, we need to rely on these buffer from the pancreas to make sure we are not damaging our intestinal lining. That's why secretin is very critical. Now, CCK, which is the other duodenal hormone we mentioned earlier, will stimulate release of pancreatic enzymes. And there's a whole bunch of pancreatic enzymes, but they're not too hard to remember because the name itself speaks for its function. So the first one is called pancreatic alpha amylase. Just like your salivary amylase, it is for digestion of carbohydrates or starches. So that's why it's called amylase, okay, which digests amylose and is released by the pancreas. The second enzyme is called the pancreatic lipase. Remember, lingual lipase? Lingual was from the tongue. Now we're talking about pancreatic lipase, and hence it is for digestion of lipids or fatty acids. Nucleases breaks down RNA or DNA material from the food that you eat, which is usually made up of cells, which has DNA. And last thing is called proteolytic enzyme, which then will break apart proteins. That's why it's called proteolytic. Protein lysis. Okay, so it's breaking apart those proteins, and usually the enzyme called proteases is what breaks large protein complexes, whereas peptidases are those enzymes, excuse me, that will break small peptide chains into your individual amino acid. Remember, we talked about how it is not until the small intestines you're breaking or chemical digesting your food all the way down to its monomer levels, and hence all these enzymes released by the pancreas will help in that process. So all of them will try to break down all those large molecules into their individual monomers. And only when they're down to their individual monomers are we able to absorb all of those nutrients into our bloodstream. So another very essential accessory organ is the liver. The liver separates into what's called the lobules. So each one of these hexagon-shaped things are the lobules, and they can contain cells called hepatocytes. So if you look at the liver cross-section under the microscope, you will see all these hexagonal-shaped lobules. You'll see that when we get to digestion lab. Remember we mentioned from our blood vessel chapter that the liver had a lot of sinusoid capillaries because there's a lot of plasma proteins and things that are moving in and out of the liver. So they will collect blood from all the hepatocytes, empty into each lobule central vein right here. So that's the central vein. And then before they can merge with your hepatic vein and go on into the vena cava. So remember, you have your hepatic portal vein that's collecting all the blood from your digestive system, and then all of those blood will go through the liver, go through your lobules, and then it will drain into your central vein of each lobule, and then merging into your hepatic vein and onto the vena cava. So central vein, hepatic vein, and then vena cava. Now remember, we also talked about stellate macrophages, which are phagocytic white blood cells that live inside the liver to protect against pathogens. So there's those again, living in the sinusoids, moving around there to look for pathogens to engulf. Each liver lobule contains 6 what's called portal triads. So a portal triad contains your interlobular vein, your artery right here, and your bile duct. So these 3 here is what's called your portal triad. Other than regulating nutrient absorption and secretion, the liver also has the important job of producing bile. So bile is an important function. Each of those hepatocytes are connected via the tiny little bile canaliculi here, where they will collect the bile that's generated from the hepatocytes, and then the bile will move into your interlobular bile duct, which will then drain into either your right or left hepatic duct, and then move on to what's called that section common hepatic duct. So bile from common hepatic duct then will flow into the actual bile duct right here and drains again into the duodenal ampulla and into the small intestine through the duodenal papilla. So That's how your bile goes through the bile duct into the duodenal ampulla and out into the small intestine. Now, if we want to store the bile, because we're not consistently releasing bile into our small intestine, when we don't need bile, it will be stored in what's called the gallbladder right there. It will flow into the gallbladder through the cystic duct. Okay, we'll get a better view of this in the next slide when I have a bigger picture of this. But cystic duct is where your bile can in turn go back to storage. So what exactly does bile do? It assists in the digestion of lipids by breaking apart the large fat droplets in chyme in a process called emulsification. This process will create smaller droplets of fat, which will make enzymatic digestion of lipids easier. Because if what you will imagine doing is, for example, if you have a huge droplet of lipid that has been sitting in the chyme as it was entering your small intestine, here's your enzymes. I always like to draw enzymes like a Pac-Man, where they are trying to access those lipids within the fat droplet and trying to digest that fat. So if it's a huge droplet, all those lipids on the inside is hard for the enzyme to get to, whereas if it was emulsified into little tiny flat droplets, then it's a lot easier to try to digest, lipid molecules from these smaller droplets. As I have mentioned, the gallbladder is for storage of bile. When bile is released into the bile duct, it actually meets the pancreatic duct. So that's why you keep seeing this picture right here, because we're talking about the liver and the pancreas, and their ducts, the bile duct and the pancreatic duct, actually merges together, and they actually go through a special sphincter called the hepatopancreatic sphincter right here in this area before draining into the duodenal ampulla and the papilla. So this sphincter here is what regulates the control the release of those secretion from the pancreas or the liver into the small intestine. And it is easy to remember which organ is secretion coming from by the name of the sphincter, hepatopancreatic sphincter. So one is from the liver and one duct is from the pancreas. Now, bile release is also stimulated by a hormone, CCK. We already mentioned that CCK is important for the function that we just mentioned on the other slide. Now, the other function of CCK is for release or stimulation of release of bile. So if there's no CCK around, bile would actually then enter through the cystic duct. So there's that cystic duct, which then, if bile was to be not used, no CCK, then it will go in through the cystic duct into the gallbladder for storage. The liver will continuously make bile. Okay, if we don't need them, they will be stored in the gallbladder. So when CCK is present, which is stimulated by presence of fat and lipid within the small intestine, CCK induces the relaxation of that hepatopancreatic sphincter. And when that is released, and it also stimulates the contraction of the gallbladder itself, and both of these will push the bile through the bile duct into the duodenum, which is the first section of that small intestine, and causing the lipid droplet, if it's large, to be emulsified into smaller droplet for easier lipid digestion. The liver actually has a lot of functions. Just those related to the digestive system already fills a whole list. Remember when we talked about the hepatic portal system, we talked about how all the blood leaving the digestive tracts needs to pass through the liver first before entering your systemic circulation. So what does the liver do with the blood carrying all those ingested molecules from the digestive system? We will talk about all these different types of metabolism in detail in the next chapter, but here is a glimpse of them. You have carbohydrate metabolism, which then if you are high on glucose, your liver will store some of that extra glucose in form of glycogen. If you're low on glucose, okay, your liver will break down those glycogen reserves and release glucose into the bloodstream, or it undergoes a process what's called gluconeogenesis that we will talk about in the next chapter. Second thing it does lipid metabolism, where it will regulate circulating levels of your triglycerides. Here is where we will talk about LDL, HDL, etc. in the next chapter. And then you have your amino acid metabolism, where your liver can remove excess amino acids for production of proteins or convert them into lipids or glucose. Lastly, you have your waste removal and vitamin or mineral storage. We'll talk about the process of deamination, how ammonia and urea is getting rid of by the liver, and then it will be filtered out through your kidneys. All right, you have your vitamin and mineral storage such as vitamin A, D, E, K, and your iron. Other than metabolic function, it also has hematological regulations, where blood is cleaned up if there are any pathogens, or remove any unused or leftover proteins that no longer need that are no longer needed, and toxins they can get rid of for us as well. So that's why the liver is also known as your detoxifying organs, or main filter for your blood. Okay, other than your kidney is a filter as well. Okay, so back to the last two parts of the alimentary canal. The small intestine is where most of the chemical digestion is completed, and products from the digestion is absorbed into the bloodstream. Remember that even though the stomach does partial digestion, it mostly does mechanical digestion by swishing and sloshing your soupy kind around, and also some preliminary chemical digestion with pepsin and your stomach acid. But it is not until the small intestine where things are really getting broken down. So if on the exam you were asked where in the digestive system are most of the chemical digestion done, you should answer small intestine and not the stomach. So just like the small intestine is divided into 3 segments. The section coming right out of the stomach is called the duodenum. Duodenum is where chyme is mixed with the secretion coming out from your pancreas and the liver, and that's why we were talking about that hepatopancreatic sphincter. That's where your pancreatic duct and bile duct merges. So that's why the first section of the small intestine is for mixing of that, those chyme with the secretions. The middle section is called the jejunum. Jejunum is where bulk of the chemical digestion and nutrient absorption happens. And the last section is called the ileum. And we already mentioned the ileocecal valve is what controls materials moving from the small intestine into the large intestine. So it will be somewhere around here where that valve is located. This section also contains the Peyer's patches that we have learned from our lymphatic system. Remember those lymphoid nodules. Okay, the surface of the small intestine is actually covered with intestinal villi, meaning that these are little finger-like projection that looks just like this. The surface of your small intestine is not smooth, but tons of little sticky things, sticky finger-like things sticking out of the surface for maximizing absorption by increase of surface area. So epithelium is lined by simple columnar cells, and these simple columnar epithelium has even more little fingers called microvilli to further increase those curvatures and adds to that large surface area of the small intestine. It is about the size of a tennis court if you were to roll out your entire small intestine surface area. The green tube here that you're seeing within this villi looking thing, okay, the green tube is called lacteal. Lacteal is a lymphatic vessel for transporting anything that can't enter into the capillaries because it's too big, which will then go through the lymphatic system and eventually drains back into the venous circulation. Now, there will be a bit of goblet cells for producing mucus that will lubricate the surfaces of the small intestine. But because it's still soupy, liquidy at this moment, there won't be as much goblet cells as the large intestine. There are what's called intestinal crypts, which is kind of like those gastric pits we saw in the stomach, except this is in the intestines. They again contain stem cells, which is for replenishing or regeneration of those epithelial cells, since again food is always moving through the small intestines and hence abrasion is bound to happen, and epithelial cells will be sloughed away and they need to be replenished. Now, Penneth cells also lies in the intestinal crypts. These are important for fighting bacteria and they are part of our innate immunity. Here's a good diagram that summarizes the hormones of the digestive system. Usually I will use this as a diagram for labeling, but the format of online quiz and online exams will eliminate the labeling portion, so you will not have any labeling during the exam. For lab practical there will be, but online exam, online quiz, you will not need to label. Now remember, gastrin is the one that is released in the stomach by your G cells within the gastric pit. These are for stimulating your parietal and chief cells, okay, so for secretion of your stomach acid and pepsin enzyme. And it increases your gastric motility when you have gastrin around. Now, secretin and CCK is also what we have already mentioned. These are the hormones released by the small intestine to induce release of pancreatic enzymes and buffers. And CCK will induce the release of bile. So remember those pancreatic enzymes we mentioned and the buffers, which is mainly for the job of neutralizing stomach acid coming from the stomach into the small intestine. The other two that we haven't really discussed yet are GIP and VIP. GIP is called the gastric inhibitory peptide, and it is secreted when fats and carbohydrates enter the small intestine, which in turn stimulates release of insulin from the pancreas. VIP, on the other hand, is called vasoactive intestinal peptide. This is for stimulation secretion of intestinal glands that will dilate your intestinal capillaries, which will then enhance nutrient absorption. Remember, all that absorption is by diffusing across into your capillaries that surround your intestines. So hence, if you have a dilation of those capillaries, you will enhance or increase the amount of nutrients that can be absorbed. So even though This diagram itself will not be a labeling question. It serves as a very good visual summary to help you remember what hormone is released by what organ and their designated functions. Okay, so use this as a visual aid in terms of trying to help you study. The last part of the alimentary canal is the large intestine, and its main function is to store digestive waste in form of feces as we know it, and reabsorption of water. That is its main job, is to reabsorb water. Again, it is divided into 3 segments. The first segment is called the cecum, which is an expanded pouch right here as a first part of the large intestine. It begins the process of compaction of things or chyme that have arrived from the small intestine through that ileocecal valve. The appendix is also coming off of the cecum. Okay, it attaches to it and serve as an organ of lymphatic system. Remember the last time we talked about it, I said the exact function is still unclear, but theory has it is that it is a storage of good bacteria. The second segment is called the colon and is the longest section. It is further subdivided into the ascending colon, the transverse colon, and the descending colon. So these are the 3 sections or subdivision of the colon. Oh, one more segment, which is your sigmoid colon right here. Sigmoid colon. So not cecum. Cecum is the first section, and then the colon you have your ascending colon, transverse colon, and then you have your descending colon and your sigmoid colon. It has series of pouches, as you can see these little pouches here. Those have a name called haustra. So haustra is sort of serving as a little pocket for segmentation of material or chyme as it moves through the colon. Now there's these longitudinal looking fibers along the colon. Those are called tenicoli. So these are longitudinal smooth muscles that will tie together and create the haustra. The last section of the large intestine is called the rectum right here. The rectum is the last expandable region where we can temporarily store our feces before release through the external anal sphincter, voluntarily, of course. Now, some major characteristics of the large intestine is that it does not have villi like the small intestine. No villi, because it's not for chemical digestion or for chemical absorption like the small intestine. But it does have large amounts of goblet cells, okay, because water is being reabsorbed as the feces is moving through your large intestine. The feces will become more dry and compact, which makes it harder to move along unless the surfaces are well lubricated by those goblet cells. Now, there exists a microbiome which contains your bacteria, fungi, and virus. These are good ones that live in mutual relationship with you. They live in your gut to break down other nutrients that weren't absorbed by the small intestine. They actually do something that's beneficial for your body instead of harming your body. So some of them actually mix vitamins. The organic waste products are your urobilins and stercobilins. These should sound familiar. Remember, we talked about this from the hemoglobin recycling process in the blood chapter, and that's what give feces that distinctive yellow-brown color. Now, when bacteria break down stuff for us in the large intestine, they will produce byproducts such as ammonia, indole and scuttle, okay? So those, along with hydrogen sulfide, all of which are not very pleasant smelling chemicals, and that's what result in that bad odor of feces, even though we don't want it to be bad odor, but it will produce that bad odor due to presence of these odd smelling chemicals. So for example, bacteria loves to feed on indigestible carbohydrates, which is sugars. They love sugars just like us. For example, from beans, sweet potatoes, they will make a lot of gas as a byproduct if you feed it all these sugars. So that results in us farting that gas all over our body. And the reason why our fart smells bad is not because we are stinky and dirty. It is because those gas coming out contains these chemicals that we were just talking about due to those bacteria eating happily in our gut. All right. So the defecation reflex involves two positive feedback loops that are again divided into your short versus long reflex. The short reflex loop is right here, and it's called the intrinsic mesenteric defecation reflex, which will trigger peristaltic contractions that move feces around from your sigmoid colon into the rectum towards your anus. The long reflex loop, which then involves your spinal cord, specifically the sacral portion of the spinal cord, is what will do the mass movement of feces from your descending colon down into your sigmoid colon and then down into your rectum and out the anus. So one is just moving around feces in this area, the short reflex. The long reflex will cause mass movement of feces from the large intestine, that descending colon, into the sigmoidal and onto the rectum. Both of these reflexes will relax what's called the internal anal sphincter, which then the feces will await for your voluntarily controlled relaxation of our external anal sphincter. Normally, it should be voluntarily done, but if pressure inside the rectum exceeds more than 55 millicurie, your external anal sphincter will involuntarily relax. Of course, this usually doesn't happen in healthy adults, but this describes what happens with newborn babies that can't control their defecation just yet. If you have taken care of or had babies before, you probably have the experience with baby poops that explode from the diaper all the way up to their backs. So now you know why. Okay, it has a lot of buildup of pressure in their little rectum before it releases those poop from the external anal sphincter. All right. So the last two slides actually serves as introduction into the next chapter, which is chapter 25 on metabolism. So don't worry about these here, these carbohydrates, proteases, lipases. We'll talk about the detail of these different macromolecule digestion in the next chapter. And this is a good diagram though that you can go back to once we have went over chapter 25 on metabolism, because we will describe each one of these macromolecules breakdown process and what waste material is generated when we try to make them or break them in the next chapter 25 on metabolism. So I will see you again in chapter 25.